Hello, my name is Jana Daoud. And today I'll be presenting to you assessing the application of CD133 expression in the diagnosis of early malignancies. Cancer treatment is a prominent concern in the medical and research fields. Identifying genes present in malignant tissue is a common approach to thoroughly understanding how disease arises in healthy tissue and how to treat it. Of these genes is prominent 1, or CD133, which is a pentaspan transmembrane glycoprotein present in neuronal cells. Studies have shown that malignant forms of these cells tend to exhibit an overexpression of CD133, particularly in your neuronal diseases. However, there has yet to be a definitive protocol in the clinical setting to utilize this biomarker as a diagnostic tool. In our literature-based study, we 1. Propose the application of CD133 as a biomarker for neurodevelopmental disease, and 2. Examine why CD133 is upregulated under pathological states. We examine similarities between the hallmarks of neurodevelopment and pathological conditions linked to CD133 overexpression to evaluate the utility of CD133 as a marker for specific neurodevelopmental states. We propose studying homologs of CD133, which is conserved among genetically tractable invertebrate organisms, such as fruit flies and nematodes as well, to better understand the reason, role, and molecular mechanism of its overexpression with the long-term goal of identifying additional biomarkers and possibly therapeutic targets. Upon analyzing several published studies, we found that in both human and mouse embryonic studies, CD133 expression is indeed highly associated with healthy stem cell and tumor cell regeneration, differentiation, and metabolism. By utilizing genetic techniques such as CRISPR-Cas9 and RNA sequencing, researchers determined that our mRNA and protein contents of CD33 are also similar in human embryonic stem cells and human cancer cells. We also found that CD133 vesicles are induced by proliferative signaling, and immunofluorescence assays can further identify whether CD133 is present or absent in differing cells. Across several cellular populations, cells that contain this gene have a significantly higher proliferation rate than those without the gene, meaning that cells that contain CD133 are more likely to not stop dividing, hence the cancerous properties. High CD133 expression is also strongly correlated to additional proliferative biomarkers such as P16, cyclin E, and Ki67. CD133 serves as a basis in the cell fate by its dynamic nature to properly repair damaged tissues or form tumors by dysregulating proliferative processes. This gene has the potential to either make the cell continue to grow and divide until it forms large tumors, or it can also healthily restore damaged tissues. Further implications for CD133 is to utilize it as a diagnostic tool to distinguish between malignant and healthy tissue. Since tissues that are malignant tend to have a very high concentration of CD133, the imaging and measuring protein assays of CD133 can help physicians sooner diagnose patients prior to the cancer reaching an untreatable stage.